I want to welcome everyone to this Juneteenth edition of the Voices in Clinical and Translational Science seminar series, which is being sponsored by the Integrated Translational Health Research Institute of Virginia, or iTHRIV. I'm Sandra Burks, in case I don't know you, and I'm the program director here for iTHRIV at UVA. The mission of iTHRIV is to catalyze and sustain clinical and translational research through collaborative team science and innovative data science and broad workforce development to improve human health and to promote health equity. This Voices in Clinical and Translational Science seminar series serves as a platform to lift underrepresented voices in research across our iTHRIV partnership, with a focus on promoting dialogue and encouraging more team science. Presenters are invited to share their outstanding research and their lived experience in science. And it is our hope that listeners will be called to listen more deeply to one another and to find new opportunities to broaden our own research in a shared quest to improve the health of our communities in Virginia and beyond. Our presenter today is Dr. Eyo from the Department of Neuroscience at the University of Virginia. Dr. Eyo was born in Nigeria, and he immigrated to the United States in 2003 to pursue his undergraduate studies at Northwest Missouri State University. He went to graduate school at the University of Iowa, where he developed this keen interest in real-time imaging of microglia during development under the mentorship of Dr. Michael Dailey. And following his PhD studies, he joined the lab of Dr. Wu, first at Rutgers University in New Jersey, and then at the Mayo Clinic in Minnesota. And in August of 2018, we were very privileged to have Dr. Eyo start his independent lab here in the UVA Department of Neuroscience with the Center for Brain Immunology and Glia, the big lab, to continue his research in microglia. He has published in diverse journals, including Nature Communications, Cell Reports, Nature, Neuron, Brain Behavior and Immunity, Scientific Reports, and many others. And he has enjoyed success in securing extramural funding, and his lab is currently supported by 2 NIH R01s, as well as some private funding. Outside the lab, Dr. Eyo enjoys the blessings of time with his family and his wife, his two daughters, and 2 sons. His presentation today is titled "The Promise of the Minority Brain Cell in Neurologic Pathology." Please join me in welcoming Dr. Eyo to the series. And Dr. Eyo, I'm going to turn it over to you.
Thank you so much, Sandra. This is an honor and a privilege to be here. I wish I could have sat in on the other previous presenters, so I have a clue of kind of how to present this. But this sort of presentation is new to me and unique to me, maybe a little emotional as well, because I'm trying to tie in some of my personal journey into my research. So I hope it goes well. Either way, it's been fun trying to put this together. Like Sandra mentioned, I've been in the States now almost 20 years, and so I'll be trying to tell you a little bit of that journey on a personal level as well as on a scientific level. This is our lab now. We call ourselves the Microglial lab because I'm not creative enough. We study these cells called microglia, and my name's Eyo, so I figured we'd put those two together. But these are the group of individuals we get to work with at different levels of training, undergraduates as well as graduate students and postdocs and technicians. We also have a lab manager. So I'll just give you a brief history myself personally. So as Sandra mentioned, I'm originally from Nigeria, which I like to think of as, if you think of Africa as a continent that looks like a gun pointing downwards, Nigeria is right in the center. It's the trigger. It's where I called home for a long time, but it's also a unique country, I think, because although compared to other nation states, it's not a very large, doesn't have a very large mass, but it does have a large population. So 1 out of every 4 or 5 African is a Nigerian. I suspect that individuals in the audience have probably met or encountered Nigerians as well. So that was home for me for a while. I was actually born there as one of twins. So that's myself and my twin sister. She's now resident in Seattle. A year after we were born, my dad, as a diplomat, was sent over to the UK. So I have no memories of this, but I have some pictures. That's myself and my twin sister. We're the last of 5 kids. There's my mom and my 3 older siblings. And I presume dad wasn't in this picture because he was taking the picture himself. So this is us in the UK. Shortly after that, we returned to Nigeria, and I stayed there for a while. We traveled different places, but eventually I ended up, as I finished college, as I finished high school, wanting to go to college. The challenge about Nigeria is that while there are a lot of bright individuals and students, the access to institutions is really limited. So we have very few universities. At the time I left, as I recall, there were 36 universities in the whole country. For a country that houses a fifth of the African population, you can imagine that that limits access to students. So eventually my dad went to India, which is where I went as well. This is me as a 21-year-old, my parents, just before I left India after applying from India to come over to the US. So then I made my transition about 20 years ago now to the US. I came first of all to Missouri. I know you might wonder why in the world would anyone come to Missouri, given that it's somewhat in the middle of nowhere. And especially for those of us outside the country, Missouri is not a state that comes to mind. I mean, everyone knows about New York, Florida, California, but Missouri is not a household name. But I loved it. I loved my experience there going as an undergrad. This is a bunch of us international students. It happened to be that there was an agency in India that facilitated students being able to come to this institution. And so this was a great experience for me coming as an international student and meeting all of these students. And I had a lot of fun as an undergrad. But I came to the States wanting to go to med school, and a little bit of my path changed in that process, which I'll walk you through in a second. But in any case, went to Missouri, finished that in 3 years as I graduated in 2006, and then moved on to graduate school, where I spent 6.5 years. And this is me towards the end of graduate school, grading papers in my last semester, December of 2012. We had just had a son who at this point was about 4 months old. And my graduate mentor came in, saw me frantically grading papers and reading papers and feeding my son, who had just woken up from a nap. And he took a picture of this and showed to me and said, Eyo, you know, there's one thing that grad school has done for you. It's giving you the ability to multitask. As you can see in this picture, you will use that ability well in the future. So I appreciate grad school for that. Then I went, as Sandra mentioned, to do a postdoc in Jersey. We had our second kid, our daughter. We had a third kid later in Jersey as we were finishing up my postdoc. And then the lab moved from Jersey to Minnesota as well. And then shortly thereafter, we moved to Virginia. And this is our family as it stands now with our 4 kids. We thought we were done with 3 kids, but we came to Virginia, and apparently there's something said in Virginia about Virginia being for lovers, and we thought we needed a lover baby. So we had a 4th one here, and we've been blessed with these ones. Okay, so a little bit about the scientific journey is that, again, in 2003, I came to the States looking fresh and didn't know what was ahead of me, but I wanted to come into grads to do med school. That's what I knew, and I was really passionate and wanted to do that. It became clear after a year of undergrad studies and looking at the cost of med school that I was not going to be able to finance med school as an international student. Back then, I think it was about 40 grand a year. And as international students, we couldn't take out loans. At least we couldn't take out loans without American co-signers. So I had a wonderful advisor who was Russian. He was amazing. His name is Peter Kondrashov. And in retrospect, I don't understand why he took interest in me. but he was my advisor, and he told us wonderful stories of research that he was doing in Siberia before he came to the States as a Russian citizen. And that just sounded so fun, the stories that he told about drinking alcohol and passing out on the microscope. And I had no idea what research was, but then he told me, Eyo, even though I didn't know what research was going to be about, he told me, Eyo, I think you'd be good in grad school. I think you should consider applying to grad school. I also had a professor called Dr. Jeffrey Thornsberry. He was my genetics professor as an undergrad student. And he just seemed so fun. He had just graduated from the University of Missouri. And I just wanted to be like these guys, seeing them and seeing how fun they made things look. And so I decided to listen to Dr. Kondrashov and apply to grad school. I applied to 3 grad schools. One was the University of Iowa. Things are different now, it seems, because students have to apply to so many grad schools today. But I applied to the University of Iowa, which was just north of Missouri where I lived. I applied to the University of Missouri because Dr. Thornsberry had graduated from the University of Missouri, and I was interested in genetics at this point. And then I applied to Washington University in St. Louis. Now, Washington University in St. Louis didn't take me. I like to believe, or I tell myself, that the reason why they didn't take me was because I submitted my application after the deadline. It may have been I just wasn't qualified enough, but I'd like to believe the former approach, that I submitted my application late. Both these 2 institutions took me, and although Missouri was interesting and desirous to me, some of the situations that our students face now was one of the reasons why I didn't go to Missouri, because the financial support that was provided was much less than at the University of Iowa. So I ended up going to the University of Iowa. Now, at the University of Iowa, again, I didn't have much research experience or hands-on research experience before this. I did 3 rotations. I rotated with a recently graduated MIT professor, Dr. Christopher Stipp. We did some cell culture. This was my first real taste of hands-on research, and I loved it. I really enjoyed it. He was a new PI, and he had what seemed like a lot of money. And so I put in a lot of work during the rotations. Our rotations were 10 weeks, and I thought I would come back to this lab and work on things. He was working on EMT, epithelial mesenchymal transition with cancer. So I thought that was really cool. I went to my second lab, which was a zebrafish lab, Dr. Diane Sluzowski, who then recently became the chair of the department. And I loved her lab. She was very interested in development. I seemed to be interested in development, but the lab seemed a little big for me, and I felt a little kind of lost in there. And to be honest, because I walked in the first lab and I did so well, I was confident that I would come back to this lab. So I really didn't apply myself as much as I could have in the second lab. But then there was a third lab, Dr. Michael Dailey, who was, at this point, I didn't even know what neuroscience was. This was a general biology department. So he was a neuroscientist focusing on glial cells, which are the cells that I study. And he did neonatal brain cultures where he cut out the brain slices and studied them. Again, because I was really interested in going back to this guy, and he didn't have much funding at the time, I kind of didn't put in as much effort. I appreciated him wanting to take me, but I really liked the cell biology that he was doing. So I did my rotations. Turned out that as the rotations were over, I went to talk to him to receive a position. And at that point was when he told me, I'm sorry, we chose otherwise and we're going to go with other things. I have later come to find out that there was a reason, which I'm not going to share now because I don't know how true it is, but there was a reason why he didn't select me. But that was a devastating moment for me as a first year, ending my first year of grad school. On the flip side, Michael Dailey took me in as a member of his lab and said, hey, you know, we're going to struggle with funding, but if you want to do this together, we can do this together. And so he took me in. I had a great time in Michael Dailey's lab. We've actually built a committee, and he was an amazing mentor, always present. Maybe I'll share one story that hits me very hard. I remember as a grad student, I told him, suddenly I'd heard that a friend of mine, a high school friend of mine, had just killed himself. And I just received the news. And I remember I didn't know what to do with that information. I just couldn't focus on anything. I walked into his office and I just told it to him. He was facing the computer. He turned around, looked at me, and ignored the computer And for what felt like an hour, he just sat there and let me speak and let me vent and let me talk. And then he gave some counsel of suggestions, but he was just present with me. And that perhaps was one of the beginning points of what I understood or appreciate mentoring to look like. He also helped me develop my committee, Jeffrey Denberg, who was an amazing instructor and teacher. Joshua Weiner, who was a new faculty member, and he was the faculty member I always wanted to be like because he made science look really fun. And he was studying, I took his developmental neurobiology class, and that was really fun and got me hooked on developmental neurobiology. And then I also had Dr. Jim Lin, who was the nicest faculty member ever, always kind, always sweet, and I always wanted to be like him. And then finally, we had to have an individual that was not part of the department, and Dan Bonthius, who was an MD PhD faculty member, was also a member of my committee. What you can see from this is that I appreciate all these individuals in the roles that they played in shaping my career development. But what is that career development in terms of its research? So I studied these cells called microglia. They are a minority brain cell because they make up only about 10% of the brain. So we are all probably familiar with neurons that are shown over here in cyan. They make these synaptic connections and talk to one another. But there are other cells in the brain called glia, for example, this green cell called an astrocyte that interacts both with the neurons as well as with the vasculature, or oligodendrocytes that interact to form myelins that wrap around the neurons. And this is important to help in the conductance of electrical activity. So while when I started grad school, and this is a picture, an image from about 2009, I don't cite the authors here because they are colleagues. And I think this is an inappropriate image because as you can see from this image, microglia are depicted here as kind of distant, not engaged with other cell types in the brain. And what my work and others in the field study microglia, and what I'd like to show you for the rest of this talk, is that they are actually pretty engaged in the brain and doing important, cool stuff, and perhaps suggest that they could be a target for neurological diseases. But they are a minority brain cell, underappreciated brain cell, in my opinion, at least back then. I think they're more appreciated now. They also are unique in that they immigrate from outside the brain and take up residence in the brain long term. I find some kinship to that myself. As I told you, I was not born in the States. These cells are also not born in the brain. They are born outside the brain in the developing yolk sac, but during the course of development, they migrate into the brain and then take up long-term residence. A third feature of them that's really unique is that they express a unique repertoire of genes. Because they are foreign cells to the brain, they do not originate like other cell types in the brain that I've shown you earlier on. And so they share a different set, they have and express a different set of genes. For example, genes that are shown here. And this is important because if we're going to target unique aspects of neurological pathology, selectivity is an important feature so that we don't have off-target effects. So these cells, once we can understand what they do, and perhaps if we can target what they do, we can potentially relieve or improve situations. And so I'd like to walk you through some of the snippets of the research that I've done so far over the last, if you will, 15 or so years, or a little over 15 years, from grad school to postdoc, and then as faculty. And I promise I'll go through this really quick so that it won't be too extensive. But as a grad student, what I had done was study the microglia themselves, a little bit of them in the course of development. As a postdoc, what I'd done was start to look at the interactions that they have with neurons. And then as faculty, one of the exciting work that we're doing is starting to look at the interactions they have not only with neurons, but also with the vasculature, which we think is important. As I tell each of these stories, I'll take a break in case there are questions, so we can interact in that way. So at the time I started grad school, it was already known and it was already done. Shortly before I started in 2006. In 2005, researchers in the field had started to ask the question, what are microglia doing in the brain? We'd visualize other cell types, neurons and different cell types. And we knew that microglia were important and changed their physical state in the context of pathology. What we didn't know was what are they doing dynamically. And we had assumed, we being the field, had assumed that these cells are stationary, boring, not exciting. We actually called them quiescent cells. That is, they were just quiet in the brain until there was some kind of pathology. Well, in 2005, that began to change when this study came out in Science, and as well as another study that came out in Nature Neuroscience that showed that these cells are not static. And if you see this movie as I played, what you can see is the cell bodies, although they are stationary, the processes of these cells are constantly moving and palpating through the brain. We now know that, or we presume that this palpation is a way for these cells to sense and feel around the brain environment and detect any pathology. So for example, if there is an injury to the brain, as I show in this movie on the right, there's going to be a central injury that we cause in the brain. What these cells are adept to do is to contain, to respond and contain that injury. So what I show here is a laser burn, and then the response of the microglial processes, in this case, within minutes, they respond and contain that site of injury. Now, there are other studies that have been done to show that if we block this response, that injury site actually starts to expand. So this is really important for the brain to protect itself from any kind of injury or bacteria or foreign material. So this was all known in 2005, just before I started grad school. But my question as a grad student was, this is known to be going on in the adult brain. What is happening in the developing brain? Are the microglia also behaving like this in the developing brain? And so that's what I set out to do. And what I did was take brain slices out of the animal at different ages from the first week of development, from postnatal day 2, postnatal day 4, and postnatal day 6. And what you'll appreciate, although I had shown you here from these movies that the cell bodies of the microglia shown here are very stationary, but the processes are dynamic. What I observed in the developing brain is that the cell bodies are actually migratory in the developing brain. So I show you that here from a P2 and a P4, and I've tracked one of these cells. You can see that over the course of a 3-hour imaging period, there's a lot of movement of the cells going on. We start to see that that degree of movement reduces at about P6, and if you get to about P9 or P12, they are largely stationary. So early on in development, these cells are more migratory rather than stationary while they crawl around and through the brain. This is all quantified here that at P4 and P6, we see a greater velocity in these cells. P2 and P4, we see a greater velocity that dips by about P6. What is important, one of the things that I also wanted to do was ask the question, not only what these cells are doing in the normal state, but also in pathological states. And the pathological state that I was interested in was developmental ischemia. And ischemia is a condition of stroke where technically all of us who are born vaginally experience ischemia, because as we come through the vaginal canal, there's some reduction in the amount of blood flow that we receive. So we all experience it, but we all do fine. Now, on the flip side, if ischemia happens in the adult brain, as we all know, probably from family members or friends, it's really dramatic. So I was interested to understand how the ischemic condition affects the microglial dynamics that we see in development. So this is one of the coolest experiments that I think I did as a grad student, where I took the brain of a 2-day-old mouse on the one hand, took the brain of a 6-day-old mouse, put them in the same chamber, and then took away oxygen and glucose to simulate a stroke and monitor what's happening to these cells. And as you can see on the left, you can see these cells at 2 days old are not affected at all. They are constantly climbing through the brain. Mind you, they have no oxygen and they have no glucose, and yet they are crawling around and tracking along. On the right, what we do observe is that these cells are not really moving at all. They're largely stationary. They're trying to move, but it seems like the ischemic condition is not allowing them to move as much. Remarkably, what I also noticed and observed was that some of these green cells, which are fluorescent cells, they actually blink off over the course of imaging. This is a 6-hour movie, and if you focus, for example, in this region, you would see this disappear, that disappear, and that disappear, right? So this blinking off of the cells suggested to us that these cells were actually dying. And this was one of the first time that we can monitor cells dying in the context of a live situation in the context of microglia. And so I was able to develop an assay where we can put a DNA binding dye called Cytox into the media. This Cytox can bind to DNA, but it requires the compromise of a cell membrane to be able to label the DNA. And we have transgenic mice that express GFP on the microglial specific promoter. So this is a microglia. OGD stands for the oxygen glucose deprivation that replicates an ischemic context. And this is 2 hours of ischemia. And as I play this movie, what you'll observe is that the cell swells up, eventually loses its GFP, and then it takes up the cytox, indicating that this cell has become compromised, it's died, and then it's become labeled. So this became a nice assay for us to monitor cell death. I won't go into all the details, but in my graduate work was what I was able to identify were some of the mechanisms of this cell death in the microglia, including the role of a specific purinergic receptor, which we now know is actually critical for maintaining ischemic health, or maintaining health of the brain during ischemia. I would also point out that it is important to understand the mechanisms of microglial cell death, because we now know from opposing sets of studies that complement each other to show that when we eliminate microglia, stroke is worsened. So when microglia die in the context of stroke, stroke is worsened in both the developing brain as well as the mature brain. And on the flip side, when we introduce microglia, we can actually improve the context of stroke. So understanding mechanisms by which the microglia die are critical. And that's something that some of my work as a graduate student started to emphasize. So before I go to the postdoc section, I can take a break here to answer any questions or thoughts so far. I can also take questions at the end.
So Eyo, I will ask one question just to make sure I'm following you. So you talked about how the microglia come in and contain when there's an injury. Are they doing the same thing if there's a virus or a foreign thing in the brain, or is it specific to an injury kind of thing? Yeah. Or is that happening with viral or bacterial presence?
Yeah, it's happening with everything. So it turns out that the microglia are the defense system of the brain. So in Alzheimer's disease, when we have plaques, they are reacting to that. In other pathologies where we have lost myelin, they're reacting to clearing that. So they're the innate immune first line of defense. Anything foreign comes into the brain or anything out of whack, they sense it, and presumably they're phagocytes, so they can clear those things out. Of course, the problem becomes in certain conditions, autoimmune conditions, they may be doing that to presumably healthy things. So for example, they may clear dead cells, which is good, but they may also kill living cells in pathological contexts when they go wrong. So being able to fine tune that is one of the approaches that we're trying to do.
Thank you.
All right, if no other questions, I'll move on to quickly through the postdoc. So this was, as I mentioned to you, my thesis committee. And as I was graduating, I used them as a resource to be able to help me to move on to the next step. I was excited about research at this point. I told you that I didn't do research before. And I should mention that when I was going to grad school, I actually wanted not to do research. I wanted to teach and not necessarily the research. And this completely flipped when I was in grad school and did all the research. And these individuals were instrumental in that. They also advised me as I was trying to pick postdoctoral opportunities. There was one in California that I could have gone to from an established investigator. There was one that was a big shot in the Glia field that I was in, that was actually in Boston. So these were big cities. And then there was the PI that I eventually ended up working with as a postdoc, Dr. Long-Jun Wu. I don't show the others' faces just so that, I have no beef against them or anything. But everybody, interestingly, he was a new PI, the PI that I ended up working with. And everybody on my committee, except my primary advisor, suggested that I go to one of these that are more established PIs. And I get that, I kind of recommend that to my trainees as well, to go to a more established setting. He saw me and knew my individual passion, and I knew that he was going to work in the space that I was very interested in. And he was the only one, my primary mentor, who recommended that I go on to join Long-Jun Wu. And so I did. And it was a phenomenal postdoc experience because he really strongly and intentionally cared for my career development. And that's partly why I'm here today. So as a postdoc, what I had done was, again, I had mentioned to you that I'd done some microglial work, studying them themselves, looking at their dynamics, as well as their viability in the context of pathology in the developing brain. As a postdoc, what I started to look at was their interactions with neurons. And so Dr. Long-Jun Wu was interested in that. And this is just one of the things that I'm just going to highlight from some of the work that I had done, which is we had used transgenic mice that have microglia labeled in green and neurons labeled in red. And at this point in about 2013, when I started grad school, it was clear that from the last couple of years that microglia and neurons physically interact with one another. But what was unclear were some of the mechanisms that they use to interact with one another. And so I used this approach to be able to visualize both microglia and neurons in the context of living tissues. And so this is a piece of tissue in the brain, in the hippocampus. Some neurons are labeled here in red, the microglia in green. And I ask for forgiveness because this was done before I was sensitive or aware of colorblind issues and individuals who may not see green and red contrast as well. So forgive me. But what we see here is that the microglia, some of their processes are making contacts with the neurons with arrows. And this is depth through the tissue, showing that the contacts are made in the same plane. What we can do is we can also drive neuronal activity. In this case, we can apply glutamate, which is a neurotransmitter that increases the interactions and signaling and communications between neurons. So in those contexts, what we saw was that there's an increase in the number of microglial contacts with neurons. We had an assay by which we can measure this. And this assay, I'll just explain it, that I developed. I'll explain it really quick. That we took a brain slice and applied glutamate. Normally, the microglial processes in green are new processes that are formed after the treatment of glutamate. So what we can see is we can see a lot of this green in this region where there's a lot of neurons present. So it's suggesting that there's a lot more interaction between the microglia and the neurons in the neuron-rich region. Then we can try to find out what receptors are controlling this. We can use a blocker, CNQX, that blocks two types of glutamate receptors, and a different blocker, AP5, that blocks one specific type of glutamate receptors. What we see is that with CNQX, we still see a lot of that green, indicating that CNQX is not able to block this interaction between the microglia and the neurons. But when we apply the AP5, we do not see any of that green, indicating to us that whatever AP5 is blocking is actually the factor that's mediating this interaction. So this is all quantified here. We can apply glutamate in this context. When we apply glutamate in the dark green, we see a nice increase in the microglial processes in that region. When we apply CNQX, we do not see an effect. We still see a nice increase. When we apply AP5, we block specifically NMDA receptors. We see that that is significantly reduced, indicating to us that the neurons and microglia communicate with each other through NMDA-dependent mechanisms, NMDA receptor-dependent mechanisms. We also asked, knowing that NMDA receptors are very calcium permeable, we asked whether this requires influx of extracellular calcium into the cell. So we did the same experiments with NMDA by itself. We can see that there's a nice response of the microglia to the NMDA treatment, but we repeated the experiments in a condition where there's no extracellular calcium. So NMDA is still added, but there's no extracellular calcium. And what we see is that there's none of that response, indicating to us that the response requires not only NMDA receptor activation, but it also requires influx of extracellular calcium. That is all on the neuronal side. Neurons express these NMDA receptors, but we wanted to ask, how are the microglia able to sense this NMDA receptor activation? Microglia themselves don't have functional NMDA receptors. So we took advantage of one of those receptors that I've mentioned to you earlier that are uniquely expressed on the microglia, the P2RY12 receptor. It's a purinergic receptor that's supposed to be able to sense extracellular purine. So when ATP is released into extracellular space, this receptor is able to sense it. And what we see is that while in NMDA-treated slices, we see a nice response. NMDA-treated wild-type slices, we see a nice response. Once we treat with the P2RY12 knockout mice, we do not see a response anymore, suggesting to us that P2Y12 is required to mediate that response. And we came up with this schematic summary of these results, showing that activation of neurons from the presynaptic terminal leads to the release of glutamate into the synapse, which activates specifically NMDA receptors and requires specific calcium influx. And then upon influx of calcium, by some downstream mechanisms, which we haven't identified yet, there's a release of purines into the extracellular space. Those purines now bind to P2Y12 receptors on the microglia, and that facilitates the microglial physical interaction with the neurons and that response. And of course, we did more work to validate this in different contexts, including in the context of seizures, because seizures is one of the pathologies where we have heightened or increased glutamate activity. So we perform seizure experiments in mice where we can either inject a drug interperitoneally, so into the interperitoneal cavity systemically to cause a seizure, or we can inject the drug directly into the brain. And what we see is that those mutants, P2Y12 knockouts that don't have that receptor, and so they cannot make that response, what we see is they have a lot worse seizures, whether we do a systemic seizure injection or a brain-specific seizure injection, we see that the seizures are much worse. In fact, we get about 40% of the mice dying in this context when none of the mice in the wild-type condition dies. So this is remarkable because although we know that seizures are a neuronal phenotype, seizures occur because neurons are too excited and are firing too much. Just by manipulating microglia, which are not neurons, we can alter the phenotypes. And some of the work that we're trying to do is to try to understand what mechanisms we can alter in the microglia to be able to alleviate some of these conditions. But that's some of the work that I did as a postdoc. And I'll be happy to take a break here before I come to moving to faculty. Do you have any questions? All right, I'll move on to faculty then. Okay, so faculty started in 2018, but before I got to faculty in 2018, there were some transitions and some things that needed to happen to me to prepare me a little bit for what I hope is a good faculty career. So I'd shown you my thesis mentors, and you may notice something that is not so unique to me was all my thesis mentors were male. And I didn't see this at all when I was a grad student because that's all we saw. But sometime about 2005, I should also point out that while I was loving research and grad school, I never saw myself either as becoming a faculty. I don't realize that I didn't see this because I didn't know anybody that was like me that was faculty. Then in 2015, as a postdoc, I went to a conference and I met this individual, Nathan Smith, who has become a mentor of mine, even though he's just a year ahead of me in his career. So he was a postdoc at the time, but at that conference, he gave a talk. And as I recall, he was the first Black individual that I'd seen give a talk. And I remember seeing him give the talk and thinking to myself, oh man, I guess some of us can give talks like this. And it actually made me, although my postdoc mentor, this is 2.5 years in, probably into my postdoc training, my postdoc mentor always told me, hey, you'll be a faculty member someday. I just could not believe it, you know? And once I saw Nathan Smith, I began to dream that this could happen. But also that same year, 2015 was a year of reckoning for me. I went to another conference and I met Dr. Michel Monnier, who was a glial biologist, a cancer neuroscientist. And I didn't know all of this about her, but she led a power hour in one of those conferences about women experiences in science, in academia, that up to this point, I was completely ignorant of. And I was so shocked sitting in a room with a number of women who were postdocs like myself, grad students like myself, and they were sharing stories and things of their experiences. And I always thought myself as a semi-aware individual. And I had colleagues who were grad students, postdocs who were women, and I just was not aware of these experiences at all. It came as if one individual had told me that, I would have been like, nah, it's just your perception. But we're in this room and there were several of them saying this, and it was just mind blown. This was one of the times that I was really mind blown and shocked by it all. So it became clear to me at that point that not having Black mentors present in my scientific career, not having women mentors present in my scientific career was not good. So I needed to diversify my mentors. I went back to Rutgers University, and I was fortunate to get to meet Dr. Bonifazi, and she has continued to mentor me and be present for me and encourage me. And Dr. Lauren Alexunis, who was the director of our T32 program that she gave me a spot, or I was on a spot on. And these individuals were very helpful to me to see what women leaders can look like. In 2016, I also met Amy Brewster at a conference. She's Puerto Rican, and she was phenomenally amazing, always believing that I would become a faculty someday as well. So much so that the next year in 2017, before I went on the job market, she actually invited me for a mock interview at her home institution at the time, which is Indiana University, or Purdue University. And the fact that she took that time out to sponsor a trip for me to visit and meet and see what a faculty talk was, faculty exposure would be like, was amazing. And I consider it an honor. And I've gone to her a couple of times telling her some challenges that I want to move out of a certain field. And she's always like, hey, you got to stay in here. You got to be in here. And it's been wonderful to have these individuals. Anytime I get a grant or an achievement, I always go back to them and tell them, thank you for investing in me and thank you for thinking of me. And this is for you as well. My achievements are for you as well. So I had to diversify my mentors. I also came to appreciate the funding agencies like the NIH doing this work to support faculty members. So I met Vicki Whittemore, who, when I was applying for my F31, I applied for it twice with a junior faculty member. I didn't get it, but she was the PO involved there and she kept encouraging to apply. I then went to a conference, and I am still amazed at this. Miriam Linders, who's with the NIH, saw me give a talk at a conference, and then she suggested to me to apply for a career development award that's specifically geared to minority students and minority trainees. I was not aware of this, but she told me about that. And then they put on a session with Dr. Michael Burton, who was a recent recipient of that award, and Dr. Michelle Jones-London, who works at the NIH to provide this. And so Michael is now a friend, and it's funny to meet some of these individuals that were distant and inspiring to you and to tell them and appreciate them. Having seen that, I decided to apply for that award, keeping in mind that I'd applied for K99 and F32s, and I'd not gotten any of those. Well, I applied for this award and I eventually got it. And the NIH has also been kind enough to put my story on the NIH website here, so you can check it out. But also, that was one step to get to the faculty position. It required other individuals to play a role. And some of you all know Dr. Yoni Kipnis, who was here at UVA. My history with Yoni goes back to grad school, because while I felt kind of isolated as a grad student studying these glial cells, Yoni came out with a paper in 2012 that blew my mind and convinced me that I needed to go into this neuroimmunology field. I started my postdoc, and then in 2014, we invited him to come give a talk. And he gave a talk, met for lunch, and he just seemed so down to earth. I always saw these individuals, these faculty members that were successful as kind of esoteric in the ivory tower, but he was just so down to earth. For me especially, he spoke about his family. I wouldn't say all the things he said because I haven't asked permission, but he spoke very fondly about his family and how he enjoys his family. what I saw most of the time in academia was individuals who I didn't know anything about their family. You know, most of them are probably not having families, right? So this was surprising to me, and he gave me counsel that I still remember of what it takes to be successful in science. And he had an acronym, HIP, which stood for being hardworking, having intuition, good intuition, and having perseverance. And I still keep that, and I still think about that. I don't know if he remembers this, but I have my own acronym that I give individuals in my own way, but he was able to show that. And so I kept in touch with him. Then in 2015, he also came out with the big lymphatics paper that was mind-blowing, and I decided, you know what, I want to come to the big center. I don't know if it'll happen, but I really would love to be at the big center where this work is going on. And then I had the audacity to ask Dr. Kipnis to write me a letter for my K award that I'd been told to apply for, and he was gracious enough to do it. I got the K award and then became faculty over here. So I'm really thankful for all these individuals who made this happen. But even having come here, Yoni was our department chair. But since then, John Lukens has been instrumental to guide and lead me and counsel. I appreciate his support. You may also know our new chair, Dr. Harry Sontheimer, has been extremely supportive as well. But beyond this, and I think this is one of the things that's hard about UVA, we don't have a lot of women mentors. And given the deficiencies that I saw in the past, I also want to ensure that I'm having women mentors present. Karen Johnson, who's involved in iTHRIV, has been extremely, extremely, extremely nice to me. And I appreciate it. I don't know if she's on the call or anything, but Karen, I appreciate you and all you do behind the scenes as well. So that leads me to the final story I'd like to tell on being a faculty member here. So this is our lab. Again, our lab is a diverse group of individuals. We're excited about studying microglia. I'll just tell you one story and one dimension of the work that we're doing, which is in the realm of the vasculature. The vasculature in the brain is really important because unlike other organs, the brain has no way to store energy. It needs an ongoing supply of energy, which is supplied by the dense network of capillaries and blood vessels. So we speculated that perhaps microglia may be playing a role here. And so this study came out in about 2021 in Nature Communications. It was work that was done in part by a postdoc currently in the lab, Dr. Kenneth Okoje. And he was able to do some laser speckle imaging of blood flow in the brain and asked the question, what if we eliminate microglia? How does the amount of blood flowing in the brain change? So this is a heat map showing a certain amount of blood flowing in the brain. And then when we eliminate microglia, we see that there's a lot more red, which is an increase in blood flowing in the brain. That is also shown here quantitatively. You can see the tracings of two hemispheres. There's a certain amount of blood. When we get rid of the microglia, we see a lot more of the blood flowing. But importantly, the brain blood vessels need to be sensitive to disruptions and changes, stimulatory changes to blood flow. So when we use a hypercapnic response by exposing the mice to CO2, we should dilate the blood vessels and allow more blood to flow. We see that in mice that have microglia, we see nice flow of blood, but in mice that don't have microglia, we don't see that. This is all quantified here that in the basal state, we get a lot more blood flowing in the brain. But when we treat with a response to dilate the blood vessels, we don't see as much. And this has led to two questions that we're trying to address now. Is in this case, we're getting rid of all the microglia and we're seeing these effects. What happens if we get rid of individual microglia on the blood vessel? What is the consequence of that? And we have people that are working on that right now. We also want to know, is this relevant for cerebral vascular disease, where we know that there's vascular dysfunction and blood flow changes, and so we're working on that as well. So I end this with some 3 takeaways so far. I hope you can get a picture of those 3 things. The first one is that mentorship and mentors are vital. This has been my experience. My recommendation for anyone involved is to choose diverse mentors. And this is twofold, so that you can receive diverse mentorship as well as you can mentor diverse individuals, people from different backgrounds. There are individuals in my lab that I don't know, I don't have the skill sets to mentor because I don't have the background. So it needs me to grow in that. But secondly, science sacrifice is inevitable. I started out with mentioning my family. This is the picture of the family in the 80s, as I showed you earlier. This is us about 3 weeks ago. We have a monthly call where we are all in different spaces. These two are in the UK. Me and her are in the US. He is in Canada. Mom is now in Nigeria. And the only way we get to talk is via phone. Thankfully, there is that, but it has required sacrifices that are painful, at least on my end, that I don't get to be with my family as much. But you got to choose your sacrifice. You will lose some things, and I hope you choose those wisely as I think about it. And then lastly, I think research is both a reward and a responsibility. I say it's a reward because I get to work with all these cool individuals. But it's also a responsibility. I have to take care of these cool individuals, as well as this family of mine, my wife and 4 kids, that give the space and the ability to do it. And for anyone doing research, see the reward and responsibility and enjoy it. And I'll be happy to engage any more as needed. Thank you.
Eyo, I'm so grateful for your time today. This is really educational for me and really inspiring, and I've loved hearing more about your story. I will start by just asking you a question to say, you know, I heard your 3 takeaways about choosing your mentors and your sacrifices wisely, and I appreciate that. And one of the things that you talked about in your own experience was looking around at your mentors and the people who were helping you and shaping you, and you didn't in that time in your life when you didn't see yourself and you had not seen people who look like you in those leadership roles and in that as the faculty role. And I wonder, what advice would you give to early career researchers now who may look around and be curious about a space where they want to be, but who maybe don't see themselves in that research environment? And the second part of that question would be, what advice can you give to this larger community to say, how can we best support those early career researchers who may be looking around and not seeing themselves?
Yeah, Sandra, those are great questions. I'm not sure I'm the right person to answer them. If after the conversation, other people have answers, I'm open to hearing that, because I think that those are 2 big questions. Maybe the thing that I would say, because I feel my position or where I'm at is that I feel like I was really privileged. I don't think I did much. I think people saw me and took care of me. And maybe what I'll say is, for those of us who don't see ourselves, be open. Don't close it off. But there are people who are willing to do all of this. And I will say this, for those of us who are minorities, there are people who are majorities that will take care of that. All the faces I showed, there are only 2 individuals that was Hispanic and Black. Everybody else was some version of white, right? So I don't know why. I don't know why those individuals took a chance on me, but they did. So maybe what I would encourage is, like, first of all, the opportunities that are coming up now that were never available when I was around. And I'm not complaining about that. I'm just saying that there are. That's more available now. So that's great. We're now having discussions on, at least on our faculty and with my chair, you know, we're having discussions on, we don't like the demographic of our department, and we're very heavily male and not female, and we got to be intentional about that. And I'm proud to say that our department in the last 2-3 years, we've hired 5 women. And so I'm saying as I see it, it's getting better. I don't want us to like take the narrative that it's always going to be bad. So based on that, I would like to tell those who feel that they don't see themselves is that there's more of us now, whatever us is, there's more of us now than there were 10 years ago. And you can be that as well. You, whoever you are, you can be that as well. But it might need you, some of my, one of my trainees, I encourage them to go to a space where they see more of them. Does that make sense? So that's what I'd advise. Anybody who feels they're in some kind of minority, if you can find your people in some sense, and you need to have those people to keep, cheering for you and that's what I would say. The second question of how can we best support? I guess we can keep our eyes open for those individuals and encourage them. Maybe also keep an eye for those individuals and encourage them, but also let's try to make our spaces more accommodating to them as best as because it's one thing to support, but if you're supporting, but the environment is not supportive, like that's insufficient, right? So let's do our best to make the environment more supportive. I'm happy to hear any other things.
Thank you.
I feel like I see Janet on the phone, and I think she's great at this.
Other questions for Eyo? Okay. Well, if not, I want to thank you again on behalf of all of us at iTHRIV for coming to be with us today. This has been wonderful. Appreciate you.
Thanks again for the invitation. Thanks, everyone.
Okay.
Happy to hear any scientific feedback as well.